So I'm here to talk to you today about SOiD, which is our open source tool to extract actions and kinematics from behavior, both trained and naturalistic. And wanted to really start this off by highlighting most of this work, almost all this work is done by Alex, who will be talking next. And so it's really a fantastic job that he has done on this, and he knows all of the ins and outs. So if you have questions, he's definitely the one to go to first. And as a breakdown for today, schedule's flexible. We'll see how things go. I'm going to try and talk for about a half hour, and then it would be useful, I think, for those of us in the ITRI lab who are going to be helping out, as well as for the general community, to get a sense of what everyone else is doing. And so, I mean, it could just be 20 seconds of what sort of data do you have, how many points, where's the camera, what's your animal or model that you're going for, and more importantly, what do you want to get out of those data with SOiD? And then Alex will give a demonstration and walkthrough, and then we'll have a breakout session so that you can work, and then we'll have several help rooms that you should have all received an email with, and I'll have a link there at the end. Go ahead, drop on in, and we have a couple people who can help you out. And then we'll meet back in this room at 4 and 4:50. If you're in the middle of something at 4 o'clock and you have to say, like, oh, I'm almost there, go ahead and skip out. But just a good chance to check in and make sure that things are going well for you. Also, a week from now, I thought it'd be good to have sort of an aftermath and see how you're doing, what questions you might have, and I'd really like to know what you came up with, because it's still relatively early days for the project.
Yeah, and I think, sorry not to cut you off, Eric, but I know that there's been a lot of interest and feedback in people wanting that aftermath session. Again, people with different levels of experience that are really, feel that they're going to be struggling for the first day. And so, we'll need to take things away, chew on them, and come back in a week. So just to make sure that that's on everyone's radar. Yeah.
And if it makes sense to have it, I just sort of pick that as, all right, a week's a good time frame. Please let me know, organize with Meaghan. Also, you should have or can get access to the SOiD Slack, so not the Department of Anesthesiology, but the SOiD Slack is really useful for interacting with other users, answering common questions or uncommon questions. So I highly recommend that as well. We're of course available to talk to. So getting into it, I wanted to start off with just sort of a very brief FAQ, particularly because most of the audience probably has a sense of what's going on with SOiD. But wanted to point out that SOiD is a statistical tool. Even though we originally conceived it to work with open field data in a mouse using deep lab cut, it'd work on a human with whiteout paint on the hand. It can work with any spatial temporal data set. And so it's not at all limited to open field per se. Similar to that, any context will work. We've had people asking about looking at breathing, facial features, reaching for a target. Someone just asked me about ballerinas, trying to get out the dance moves. And then in a sort of side project, Alex is working on a project to actually extract out signs from American Sign Language. So it's very versatile, and there's also various levels of user interaction. We designed this to be essentially plug and play, given a good marker data set. But if there is a particular behavior that you're looking for, a particular subclass, you can actually program that in, and SOiD will integrate that. so one of the big questions is, why even have this? We've been doing a good job in neuroscience being almost exclusively reductionist in our approaches. And here is data from my own lab with a mouse doing a reaching task, moving the joystick up and down or side to side. This is great. It's learned this behavior, but we have all sorts of questions. One, mice don't typically come across joysticks when they're going around in their normal everyday life. They're not evolutionarily designed for this. And also, this animal has undergone thousands and thousands of training trials. So we might want something more, more concrete to the animal. Also, training is just a pain in the butt. So with that idea, there's been a growing urge to look at naturalistic behavior. And of course, BSI can be applied to any set of behaviors, even trained behaviors. But one of the ideas here is that by studying naturalistic behavior, we're getting at the brain's innate state. So no training is required. There's no confound from overtraining. But the naturalistic behavior still is a problem. There's a difficulty to classify what those behaviors are. How do you parse them out? How do you deal with variability, which is one of the biggest reasons why we haven't really switched to naturalistic behavior so well. It's easy to establish a trial structure, but when you're not enforcing rules on that, the trial structure goes out the window. And so we sought to overcome these issues, largely in response to the growing technology that is computer vision plus machine learning, specifically Deep Lab Cut. LEAP is another program. There's some other algorithms out there as well. And those are fantastic. They're really great. I would not want to put markers on every single frame. That'd just be pretty terrible. But there's still a problem. It only gives you positions of those limbs, the poses. And this isn't behavior. It's the spatiotemporal relationship of these positions that gives us behavior. Things like grooming, walking, rearing, reaching. And so we needed a way to extract the natural statistics from these points as very compressed feature space. We're not dealing with a whole video, but to get the relationship both in time and space. And so Alex and I, again, largely Alex, came up with BSI. And here we have mouse navigating an open field, cameras from below, so we get a good access to all of the limbs. Also, it enables us to have the animal tethered if we want to. We don't have to worry about any cables getting in the way of the video. We see here a projection into t-SNE space of all of these identified actions. And it's important to note, we're only assigning the names of these actions, these behavior groups, after the fact. The algorithm has no idea how many there should be, has no idea that there's a face on this animal. It's just whatever the naturally occurring statistical clusters are. That's what's going to be pulled out. I want to take a quick break here before we get into the good stuff. I imagine everyone's quite familiar with this stuff so far. Any questions? I'll take the silence as no. And if there's something in the chat, I can't see it. I don't think I can. So go ahead and speak up if there's something in the chat.
Yeah.
Go ahead.
Sorry, Eric, there was one question in the chat about not having a data set of their own to work with. I just messaged them privately and said that's fine. They can still participate in the webinar.
Yep.
And then Booma has a question. How are the action dimensions defined?
We're about to get into that. Good question.
Yep. Can I ask a question as well? Sorry.
Go for it.
I just wanted to know, because obviously the way that it's plotted, you lose like the temporal progression of these events, right?
No. So even in this, we can see each one of these dots is placed in time as it occurs, and we synced up the video. So what BSI will output is whatever group number a frame belongs to and the time of that frame. So you get a temporal progression there, and it's using both the spatial arrangement of those features and the temporal progression of and by features I mean the positions, the output of Deep Lab Cut, in order to arrive at these classes, these groups.
So afterwards, you could still plot all of these a long time and still get the same groups.
Yep,
Okay.
and I'll show you that on this figure here. These are positions and then the segmented behaviors. But first, just an overview of the computational process. Alex will get a little bit more into this, but I think focus today will be largely more on user experience rather than the computational side. But the idea is that we want to go from just the points in space that Deep Lab Cut or LEAP or whatever our marker system gives us to extract high dimensional features such as length, speed, angles between the points. And then we use t-SNE to find those clusters. And so this is we're compressing across dimensions here in order to find the clusters that may or may not exist. And so D up here has an example of one of those field of dots. Each dot is a separate frame. And something like this had been done before. Rui Costa at Columbia had done something like this before. And this works, but it's still a little bit rough. And also, you can't have the ability to compare across animals or even within one animal session to session because it's a stochastic process. You'll always get slightly different groupings. And so the idea here is to use these data rather than just having this statistically clustered behaviors as our output. We use machine learning. So we take this information to train a multi-class classifier, an SVM, with these data and says, all right, if there's a distribution, and here we see an example of distribution for face grooming, body licking, orienting, and here's some of our high dimensional features which have been conserved throughout the whole process. We can train the SVM in order to go through any data, an unlimited amount of data after that point, to go through and say, all right, this is group 1, this is group 7, this is group 5, because it fits to these groups, to these distributions. Interestingly enough, the machine learning version actually outperforms the other just using the t-SNE. That's largely because the machine learning can make inferences. The statistically clustered behavior, it only has access to the information that's there, whereas machine learning is making inferences and can thus better classify these groups. And so what does it look like in reality? Here we have some of the clustered behaviors in brown and black mice. Really doesn't matter because, again, it's the Deep Lab Cut or the marker data that's going in. So as long as you can get quality data, there's no problem with different colors of mice. And we see great accuracy, particularly for the Gaussian kernel that we're using. That's this cross-validation is maybe a bit beyond what really the purpose of this webinar should be. But happy to get into that, or Alex can as well. And then the real question is, how well does it perform? So on top here, we see that throughout time, here's one transition from a rear to a head groom, and then from a fast locomotion to a nose poke. And we see left and right limbs as blue and red. The forepaws are in darker colors. And this is just the kinematics. Again, another nice reason to have a camera from below. You can get these kinematics as well as behavior, but you don't need to have that. And we see that this isn't bad. It's still, the accuracy is pretty good, but that the machine learning algorithm improves upon this because, again, that machine learning algorithm is able to make inferences. But this still isn't that great. It's still only about 100 millisecond resolution. So we took one more step, and this is about the, if you have a commercial system, you're doing at best about this 100 millisecond resolution. Or if you're using MouseSeek, Bernardo Sabatini's algorithm. But we wanted better. If we're going to be recording neural activity, we want to be in the millisecond range for resolution. But there's a problem with that. If you're doing high sampling, the jitter of just your technique will always be slightly imprecise on how that point is assigned. And then there's just jitter in the signal in general. If you're oversampling, the signal that you get will be largely jitter, not the actual movement of the limb. The jitter itself will predominate. And so we needed a way, so if we want to really get, in this schematic version, a movement from this point to this point, we need a lower sampling rate to resolve this, to get the signal above the noise. And so to do that, we took a high-speed video, but then downsampled it, which allows us to get this nice characterization of going, for instance, transitioning from behavior A to behavior B. But in order to recover that temporal accuracy, we then took that downsample and shift it, and then shift it again, and then looked at where the average transition went from behavior A to behavior B. So instead of a highly sampled set of frames, we take downsampled, but now we can shift it at the frame rate of the camera. We recover the original frame rate. That's been a great improvement for this. Here's just showing we make the decision as to when more of the samples are in group A or group B as the official time of transition. And here's our pretty good performing machine learning based algorithm. And with the frame shift, we get really clean temporal resolution. Want to take a break for more questions if anyone has it? I'll assume that's a no again. Again, I can't see the chat.
Sorry, there is a question in the chat. I could read it if that's fine.
Sure.
So it's from Gerardo again. Do you guys ever see this working in real time for a feedback system, or can it already? Could they be rewarded for grooming in a specific position, for example?
We are definitely interested in that, and that's one of the reasons why we've really pushed on this. I kind of, my postdoc got a Nature paper for real-time closed-loop feedback stimulation during a particular type of behavior. That's very much in my wheelhouse, and we're aiming to go towards that. Deep Lab Cut is only just now coming up to speed with something approaching real-time. When they get there, B-Side should be able to do it quite quickly. We're working on, real-time is sort of a relative term. Real-time in terms of what would be useful in an experimental setting, we're approaching that. So it does not exist particularly in any well-tested form yet, but that is certainly on the horizon. All right. So we've just been looking at these kinematic readouts, which being able to get the behavioral classes is pretty fantastic, but wanted to show an instance of where this high temporal resolution kinematics can actually help as well. So here we have a face groom, and just wanted to take a second. This is more for the big neuroethology behaviorists in the room. Again, B-Side has no idea what any of the groups are, but I thought it was pretty fantastic that there's 3 canonical types of grooming in the literature, and BSide pulled all of them out specifically. So that's pretty exciting. Anyways, back to this. In a face groom, we took a normal animal and an animal in which we had used a caspase virus to kill all of the indirect pathway projection neurons from the striatum. This was done by Erin Giddis's lab. She gave us the mice. And we found that for face grooming, that there was a very clear difference in the performance of this face groom. And importantly, not in the dimensions that most people look at. If you read, I know we have several people who study pain and OCD and itch in the room. When you study grooming or itch, you're typically looking at bout duration. We saw no effect between the 2 groups. But when we look at the trajectory distance or the speed that's happening at, because we have resolution at the frame rate of our camera, I believe in this data set, it was at 200 frames per second. It might have only been at 100 frames per second. Either way, quite quick. We were able to see this very, very clear distinction between the normal and lesioned animals, and particularly down here the bias was very specific to the smaller speeds and the smaller distances that really find behavior, which really is even more important when we look at a larger type of groom, which again, several groups are not going to make the distinction between these 2, just because it's so difficult to quantify if you're scoring by hand. With these bigger movements, there was no effect in any of the measures. So this just really speaks to the importance of having not just a great ability to parse behaviors, but to get those kinematics as well. And your mileage on kinematics will, of course, be limited to what you have marked in your own data. I can go through this, sort of interesting because we can get these behaviors and throughout time. We rediscovered a phenomenon of just behavior changing in time. We looked at an animal in the first 30 minutes in a novel field versus the second 30. We found 2 processes going on. One, similar behaviors. So we grouped our behaviors as just all the grooming together, all the locomotive behaviors together. They all grouped in this feature space. This is a spring graph. So that didn't have to happen, which is sort of interesting. But over time, we found there was a change. The probabilities of transitioning from one behavior to another became more normalized. The high probability transitions became less, and the lower probability transitions became greater. But the overall structure was largely maintained. And this can point to future experiments wherein we might look at the decision-making process underlying this action selection throughout time. Just sort of something cool that we can pull out with these approaches, but in this case, it's not necessarily specific to BSide. So just a quick summary of what we've gone through. One prompt gets you all behavior from any marker system. It captures limb position if you're marking it, unless it provides actual kinematics as well as behavior. It is, I didn't hit upon this as much, but because it's agnostic and unsupervised, there is very little room for experimenter bias, which is fundamental. We might think, oh yeah, a mouse thinks of that as a scratch or a fast and a slow walk. But BSide is actually providing a mathematical truth. Still not entirely sure what the mouse thinks or the monkey or whatever you're studying, but there's some truth in the inherent statistics of that behavior that we're able to pull out. And that in this case, the animal can be tethered, and it will really work with any spatial temporal data set. just wanted to get into a trained behavior, different species, different tasks. Here we have data from Dan Leventhal's lab that's on the original deep lab cut paper, where a rat is trained to reach for a sugar pellet, and they have several markers on the pellet and on the hand. We took this data and we were able to, we just wanted to see what we would find. And here's a successful reach and one failed reach. And we see that deep lab cut, or sorry, BSiDe is able to pull out from deep lab cut several behavioral subcategories throughout the trajectory. We line up all of these reaches over time. We see that these subcategories, these subactions, are very stereotypical or very stereotyped. It's not just randomly assigned. And these are only for successful reaches. Failed reaches come in many varieties. In this case, the mouse goes past it and reaches around for the pellet. But we can categorically see just from these subactions whether or not a rat was successful in their reach. Here's one group that was pulled out. Again, BSiDe has no idea what the data are except just where those points are. Here are two versions of a failed reach. In this case, there was a pellet that was missed. And in the other case, the pellet actually fell off the pedestal. So it's reaching for nothing. And I want you to look at the middle digits. Let's look at that one more time. So it's that splaying. And that BSiDe was able to yield that as a category and get this type of in this case, type of fail, which pretty impressive to me. So wanted to wrap up this sort of overview section and happy to take any questions at this point. Here's sort of a summary of the state of the field, as it were. So be happy to take any questions. That's a pretty easy question period. So if there aren't any questions, and I'm sure we're going to learn more in a bit from Alex, I'd like for everyone to go around and tell us what model system, tell us about your deep lab cut data, if you have it, or LEAP, or whatever the pose estimation is. And really more importantly, what you're hoping to achieve with BSiDe. And